All right, I think we're almost at the end of day one. So this is our last breakout session for today on the patient's role in drug discovery. We, of course, have the live stream folks from Platform Q. So just to remind everybody, there's remote access. The last session of the day is Dr. Deborah Requesens from Cornell Institute for Medical Research. She also serves as the co-principal investigator of the National Institute of General Medicine Sciences Human Genetic Cell Repository. That's a very long title. She will be providing insights into the world of biobanking and what the best ways are to contribute to these databases. So I'd like to welcome her up on the stage. She'll give her talk this afternoon and take questions. And then the last part of the day at 4:45, just to remind everybody that we have Dr. Philip Riley as our special guest speaker, who will be giving his talk this afternoon, followed by a book signing. So with that, I will introduce Dr. Requesens.
Hello, good afternoon. My name is Deborah Requesens. I am the program manager at Cornell Institute for Medical Research, and I'm also the co-principal investigator of the GMS repository. It is a mouthful, but it's the National Institute of General Medical Sciences. I want to thank the organizers for inviting me today here, and thank you all for being here and listen to this talk. That's what it is. Today, I'm going to give you some more details of what a biobank is, including the types of biobanks that we have. I will give you some details of why people should donate to a biobank and how to do this. We will talk a little bit about the uses of the samples once they are stored with us. And finally, I will finish with some idea on how to create a private collection without a biobank. So what is a biobank? The general definition of it is a collection of biological material that was set aside for research. That would be like the general this definition of it, but there are many other small ways to define it. Many people call it a library of biospecimens, and I'd like to add that they are appropriately stored biospecimens, since that's actually the main role that we have at a biobank, is how to store them. Hopefully the samples are of high quality and hopefully well characterized. They can be cataloged by many ways. They can be cataloged by type, species, genetic traits. They are also hopefully easily and affordable to scientists all over the world. And without a doubt, a biobank is an invaluable resource for research. So what is in a biobank? Most biobanks have blood, cells, DNA and RNA that's been isolated from these blood or cells. That's what we have in our biobank. But we also can have saliva, plasma, tissues, and many other bodily fluids. So how are they stored? They're mainly stored in cold. They're very cold, either liquid nitrogen tanks, as you see in this picture. They are at minus 316 Fahrenheit, or some of them are stored in freezers that are -4. Some biobanks will store some samples at ambient temperature. It's not our case, but it is possible. So what other types of biobanks? They can be defined on either population-based biobanks or disease-oriented biobanks. The population-based biobanks are those where the samples have been stored and collected and stored from different particular geographical areas. The most famous ones are the 1,000 genomes, so the International Health Map. This morning, Dr. Goodhand actually mentioned these two as the most important ones. They are stored at the Cornell Institute. The other type of what I'm guessing most of you are being interested in today are the disease-oriented biobanks. And this can be either cells and DNAs, like the one that we have, the rare disease one, or it can be tissue. The tissue biobanks, I am not an expert on this, and we do not store banks, but I know that our genetic counselors, when they are contacted by families, usually refer them to either one of those two, the National Disease Research Interchange that has tissues that are not suitable for transplant. Or if you have brain tissue, the University of Maryland Brain and Tissue Bank, that is an NIH-sponsored biobank, is the one that would refer to. So I'll be glad to give you contacts about them, since it's not what we do. So what is it that we do? We do have cells and DNA. We are at the Cornell Institute, and we have these cell lines and DNA that are well characterized and high quality. We're very adamant that our cells are of high quality. Our goal is to stimulate and facilitate biomedical research. We want to facilitate teaching and diagnosis and prevention of human genetic disorders. We are independent. We are not-for-profit. We are actually funded in big part by the NIH. We distribute and collect samples all over the world. And we have very diverse different collections, either for disease collection populations or species, like I said earlier. One of the main and most diverse collections that we host at the Correal Institute is the NIGMS repository. Again, this is the National Institute of General Medical Sciences, and our repository is called the Human Genetic Cell Repository. This repository was funded and still is by the NIH since its inception in 1972, so it's been with us for a long time. We have over 11,000 cell lines stored in our biorepository and almost 6,000 DNA samples. These samples represent over 1,000 genetic diseases, but we also have chromosomal abnormalities, what we call apparently healthy individuals. These are usually the family members, the blood relatives of the samples with the disease. And we also have stem cells, and I'll tell you a little bit more about that in a minute. We distribute to all over the world. Just last year, we sent samples to over 70 countries around the world. And we are proud to say that we have over 7,000 publications linked to the samples that we have in-house. There are many more, but that's a very arduous task to actually link every publication that comes out. But we try to keep up with the publications that are out there using samples that we have. So why should you donate a sample to the biobank? The main and easy answer is because scientists need these samples to advance research. In fact, a survey that came out recently that asked about 700 investigators, 40 of them said that they could not find the samples that they needed, and 81% of them said that this affected the research that they did. It's easy to understand that a scientist that wants to do a study with cells or DNA needs to, first of all, find the people. So we're talking about rare diseases. They are all over the world. There can be just a few in one place. So it is very hard to, first of all, find the patients, then have everything that has to do with the paperwork, IRBs, consent. So this is a lot of time. And then they have to set up the cell lines and establish them. So the idea is for them to find everything they need in one centralized location. And that's what a biobank does. By using a biobank, the investigator can then find the samples in a quickly and affordable way. They can, at least in our case, we guarantee a high quality type of samples, and they're usually well characterized. And again, the samples can be from anywhere in the world, and we can ship them to any place in the world. So who can donate the samples? Samples can come from anybody with a diagnosed genetic disease or a chromosomal abnormality. In this case, this is a very famous picture of our friends, our Phi NPC, holding the kits that they received from Correal and being very proud of their skin punch of the samples that they sent. But also we accept samples from the close relatives of the affected individuals. The ideal is to have the trio, to have mother, father, and proband, but also siblings are accepted. How do you donate your sample? Well, the first step is to contact us. Here's our email, but I have a few brochures and cards if you're interested for your group. So just to contact us and make sure that the disease, it is a genetic inheritable disease and that we are accepting at the time. And we'll be happy to send you the kit for the collection of blood or skin. Once you receive this, we ask you to complete forms, including informed consent and submission. And we do request that you send us as much clinical data as possible. It is important to have as much clinical and genetic data accompanying the samples. For the research community, this is a very interesting thing. The more data that the sample has, the more interesting it is for the research community. After this is done, just get your blood drawn or the tissue taken. We recommend to do this during a very routine visit to your physician. So in that case, we can send the kit directly to the physician. So we don't have to send it to you, we can send it to somebody else. And once everything is finished, you put it in a box that is a prepaid box and send it back to us, and that's that simple. It's all free of charge. What happened to these samples then, once they are in us? And this is probably the most important part that I want to talk about today, is that the samples do not stay with us all the time. We don't want it to stay with us. We want the samples to go somewhere else. We ship to every state in the United States, and we ship to over 70 countries, as I said earlier. So think about the fact that these samples that get to Camden, New Jersey will not stay in Camden, New Jersey. Hopefully they will go on any other place. And what happens when the scientists get them? Well, so many things, from basic research, this is research. They can discover new genes, find out the function of these genes, can develop disease treatments. They're developing genetic testing. They're validating and doing proficiency testing. And they're generating what we call man-made stem cell lines, or induced pluripotent stem cells. This morning there was a lot of talk, and I think Dr. Ring mentioned this a lot, but this is a very hot topic at the moment, where you can take a piece of your skin and then make it a stem cell, and from then differentiate it in any kind of cell. We're working a lot on this, on what we call disease on a dish. It'd be great for clinical trials. It'd be great for trying and testing therapeutics. So we are very much on top of that. Many of our scientists purchase our cells for that purpose. We also host some of them in our catalog. We either reprogram it ourselves, or we get it from other scientists, but this is a big part of what we do. So once that I tell you this, of what happens to your cells, I wanted to tell you about a few concrete examples of who works with us, and different organizations and rare groups that have done collaborations with us and what's come through them. So I'm going to tell you about how our samples sometimes are used for their purpose, to help out the disease. Sometimes they've been used for something completely different and unexpected, and how some people are just starting with us and they really want the community to donate samples to us and take it from there. So I'm going to start with what I would like to call the NPC Ebola link. We receive a lot of samples from NPC, and probably some of you know the Niemann pig type C disease, lysosomal disorder, is very devastating. And we received at some point many cells from affected individuals with this disease, and they were distributed. One of those samples, so some of those samples were distributed to Dr. Chandran in New York, and they found something completely unexpected. They discovered that the cells with the NPC gene, the defective gene, could not be infected by the Ebola virus. And we have a video, and I'm going to let him tell you what is it that he did.
We were able to get these Niemann pig patient cell lines from Coriell. And so these cells are actually really useful because they allowed us to really understand what Ebola needs in terms of NPC1. We're really using the virus as a tool to teach us about exactly how NPC1 moves around in the cell in order to do its job. For example, what the structure of NPC1 looks like as a molecule. Having the structure of it would allow you to develop better drugs to preserve its function. We're doing this from this virus-centric view, but I think that this general knowledge that we would learn about NPC1 and how it works by really using the virus could, provide information that would be useful for other scientists that are trying to develop therapies to treat Niemann pig disease. And so, in fact, we have a collaboration with a scientist at Mount Sinai, Yannis Ioannou, who works on NPC1 and Niemann pig disease, where they're looking for drugs to make NPC1 better. But in the process of finding those drugs, they also found drugs that make it worse. And so we're taking those off his hands and using them for Ebola. And so if we go looking for drugs that make NPC1 worse, we may also find ones that make it better, and we can give them to other people. We're hoping that the work we do will benefit, the work that's going on to treat Niemann pig disease in just the way that the work that's being done to treat Niemann pig disease has already benefited the work that we're doing.
So what I hope that you heard what he was saying is the idea of sharing and collaborating. With one type of samples, the intended use was to do research for the NPC1 gene and develop treatments, and that's happening. But also we are now researching and developing treatments for Ebola and other viruses, which was a completely unexpected outcome of these studies. And this might sound kind of funny, but these are the twins that we believe the original their cells were the ones sent to Dr. Chandran, and their mom, during an interview, mentioned this. She said, my first thought was, holy cow, followed by a strange visual of ourselves floating around in Petri dishes with Ebola virus and people suited up in hazmat suits. This was very interesting to us because it showed the fact that people don't think about their cells living once they are with us. And this is interesting to know that it's important for me to actually people know that they do. So they do fly. They actually literally fly in airplanes all over the world. And sometimes they go with things like Ebola virus. So this was a very, very interesting thing for us to share with us, with everybody. The next step is the one where what we expected to happen happened. And this is the link between the Choroideremia Research Foundation for the molecular therapeutics. Choroideremia is a progressive death of retinal cells that starts with dark blindness in children, and it causes total blindness later on. Many cells were donated to us with this disease and then purchased by different investigators. The investigators at the University of Wisconsin have been working with the cell lines, making human pluripotent stem cells and other type of cells. And also for the molecular therapeutics, have purchased our cells, have been working with them. And they were very happy to announce in February this year that they are developing a gene therapy for this disease. So this is a very great example of how the use of these cells in one centralized location has been created a collaboration and hopefully a treatment for this disease. And the next case that I want to talk to you is our NGLY1 case from the Grace Science Foundation. This is a disease that is very, very new and very rare. The lack of this enzyme just makes that the misfolded proteins are not recycled, so they're accumulated in the brain. It's very devastating, and it causes all sorts of things. And I should have said hypotonia in there. So there's a typo there. I apologize. These are the two kids that are kind of famous for this. I don't know if you've heard about Bertram Might, but his father, Dr. Might, is here, and we'll be talking tomorrow. So I recommend you go and listen to his talk. But their mutation was discovered by whole exome sequencing, and they, especially the Wilsons, which are the family on the right, their Grace parents, they got a lot of people asking them for cells. And they did not want to keep having to have their kid taking their blood and their skin. So they started the Grace Science Foundation, and I'd like to read something that they say on the website about biobanking. They say it is important to streamline the way scientists procure tissues and cells. And then he's talking about our institute, and they say they allow researchers to quickly and easily get tissue and cells without requiring us to coordinate shipments or go back into the lab. So that's a very important point to take, that the way we do this, you people don't have to continue to give their cells and give their blood. It will be in one place, you tell your scientists what it is, and that's where they go and get it. They also say at some point somebody said that their cells weren't growing, and he said, Korea could have fixed that. And we're very proud and very happy that he actually said that. We do proud ourselves on having high quality cells. Other than these particular 3 cases, I have to say that every lab that we work with that has been studying any kind of treatment, they all get samples from us. Any little lysosomal storage disorders in particular, they are what we call our top sellers. We distribute a lot of these cells. And every small lab that is working on this has to get their cells somewhere, and they get them from places like us. So it is very powerful. Also, not only the disease cells are used in studies, cells from our population groups are getting things like studying the flu vaccine, discovering genes for other diseases. We also are very involved into the reference materials. And this is something that is very hard at the moment but I don't know if you were here this morning when Dr. Taft was talking about sequencing and how sequencing is the new thing. Well, you have to think that anybody like Illumina, the company that he's with, or any other company that does sequencing, needs to compare the results with something as a reference. This needs to be done to be able to improve accuracy, reliability. Then we need to reduce the false positives and the false negatives. So the use of these reference materials is very important. They just came out that our NIST, which is our country's institute for standards, just came out with a new set of reference materials this past Friday, actually. And the samples that they're using are hosted at Cornell. So this is a very important use of cells. And I want to finish today by talking to you about private collections. And I don't want to start by saying that this is not something that is necessary. This is just for groups that have the funds, first of all, to do it. Everything that I mentioned before is free, since the NIH is funding them. But some people have some concerns about what's happening to their samples and where the samples are being distributed, and they want some more control of what happens. So a private collection might be the right move for them if they have the funds. How we can help them, we have over 60 years of experience. We can establish your repository. We can do the banking, the preservation, the storage. We have a very complex system of informatics that can help with the whole process. We have an online catalog. And again, I can have some brochures if you want to look at them, where scientists and researchers can easily find the samples that they need. We can collect and distribute all over the world, and we have a very good plan for disaster management. It is a very complex and time-consuming process, so I'm just going to guide you a little bit of what happens and how this is done. First of all, the sample collection. We do provide the kits, so we will send you everything that you need for collecting either blood or skin punches. Then once you collect the samples and you fill out all the informed consent, we will guide you through that as well. This is sent to Coriell, where we will process your sample and do all the quality control. Once this sample goes through all the control, it will be on a catalog online where your researcher can come and look into the catalog and see what they need and order from it. We will then ship this back to the scientists and do all the reporting for them. This is just the same kind of idea, but just in a more general way. So just to show you how integrated the whole process is. On one part, you do collect the samples and send them back to us, but we do the collection, the accessioning, the quality control, and storage. Then everything goes into catalog and gets distributed back to your scientists. This gets a lot of people involved. It's a more complicated process than we think. It has a lot of project management. We have very high-tech technology laboratories, cell biology and molecular biology. We have a big group of IT that takes care of this. And we have, of course, a lot of scientific expertise. And the most important thing is that many people like about this is that it can be completely customizable. It's whatever you need and however you need it, so we can work with the group. And finally, I want to finish with an example of a private biobank that is within Coriell. And this is the CHD Genes Biorepository. This stands by Congenital Heart Disease Genetic Network Study. They are a group that is studying a congenital or acquired heart disease in children, and their goal is to identify their genetic causes. So this repository collects blood, saliva, and DNA from children and their parents with this disease and distribute it back to researchers. So how do we help them do this? We provide all the collection kits. This is a saliva kit that you see here on your right. But we also provide the kits for blood collection and also sometimes DNA. If what we receive is blood or saliva, we extract the DNA from them, do all the quality control, aliquot it the way that they want. Some people, at least in this case, they require the samples to be in special plates, so it takes some work to do that. It's very what they need for the labs to use, and then we send it back to the researchers. We do all the tracking, we generate reports, and we collaborate very closely with both the CHD genes and the researchers. So as you can see, if you have the funds to do something like this, it can be tailored to your needs. And with this, I want to finish with a phrase that I heard recently in another talk, and somebody said, please don't store your samples in a biobank, collect them for sharing. And I think that's the most important thing that I want you to take with you, is that these samples will not stay there. They will be shared and utilized. Thank you so much. If you have any questions, I'll take them now. Sure. Here's your microphone.
Hi. You had mentioned this last one, this group. Was that privately done, that they're paying for it then,
Yes.
that group?
Yes.
So for a particular disease, either the patient advocacy group or the family will have to store the samples?
If you want something that is that particular, yes.
To that disease.
In this case, yes, because it's a completely, they have their own IRB, they have their own collection places, and they only distribute to their scientists. So yes, it is funded by the group.
Got it. Thank you.
You're welcome.
Two questions. Do you, of the 7,000 some rare diseases, how many have you accounted for in your organization?
We have almost 1,900 and something, and 60 or 70.
Okay. And do you solicit for more, or do you just stick to those?
Oh no, we want them all. We want them to be, we want scientists to be able to find everything they need.
Okay.
So yeah, so we're open to accept. The more rare, the better.
And since you're funded by the NIH, do you ever supply the NIH with samples?
Why the NIH?
Do you supply them with
We don't know.
samples?
They just fund us to do our
They just
job.
fund you? Okay.
Yeah. We do, of course, have a scientist from the NIH come in and get samples, but they're like anybody else. I mean, they don't go through a special process. They order the samples through catalog like anybody other researcher.
Hi, I have a question. My daughter has a very rare syndrome. If we decided to give our samples to the biobank, would we be notified if someone wanted the sample?
No,
So you don't
the
find
way
out?
the NIH has decided their IRB is that the samples are completely anonymized. Where they're not linked between if you do it through a researcher, the researcher might get more information about the samples, but there's no direct contact with the patients.
Okay, and then a follow-up question to that. How many researchers, like if we sent in one sample from my daughter, would that go to multiple, or would that one sample only get to go to one researcher?
Oh, it will go for as many people. These cells are immortalized. So they will be with us forever and ever, and they can be distributed to anybody. And actually, if you do send us once, you do get a free back to submitter, so the researcher of your choice will get a free sample back.
Great, thank you.
And I have a brochure. I can give it to you before you go.
You mentioned that researchers would go to the catalog and order. Do you know about what the cost is associated with that?
Sure. It depends. We do have a different pricing for nonprofit or academia and for private labs. It goes anywhere between 50 and 85 for. Sorry, she has another question. For DNA sample, and 85 to 100 for a sample of a cell.
And
So it's very it's what costs us to make them. It really is not, and not even because we're funded by the NIH. So it really just covers the minimum. We're nonprofit, so.
and are there rules as to what researchers can purchase these cells?
Well, we want to make sure that it is an official institution. We do not ship just to anybody that requests ones. We do request also a form that explains to us what is that the research is going to be done with it. We also request a lot of paperwork detailing that the cells will not be used for commercial use, only for research. The knowledge from the research can be commercialized, but not the samples themselves. So we do have a lot of regulations, yes.
Hi, can you explain again why people would take the private collection approach? I mean, what's the advantage of doing that?
In the case, for example, of the Ph.D. genes, they work with a particular group that they are funding themselves. So in their case, they just wanted the samples to go back to them. In my personal opinion, I think the more researchers that touch your sample, the better you are. That's also my personal point of view. But in this case, they really wanted to control who was getting back the samples because they're getting results from these scientists in particular for their group. So it's a personal opinion.
If a researcher finds information about a particular person from a sample, can they come back if you authorize them to?
We try, unless the sample has been agreeable to, like the donor has agreed to have their personal information displayed, it is not. And that's one of these big hot subjects about data sharing. A lot of our samples are not accepted to post any kind of personal information.
So it's all de-identified information?
It's all
that goes-
de-identified, yeah, completely.
So they can't, if they find some big breakthrough, they have no way to come back to tell you?
No, it's not the way that this was set up. You can probably contact us, and we could, if it's something that was, I can't even imagine, but how we would contact back the researcher. But it is made in a way that these samples are for research and they're not for a personal and individual's gain.
And you also mentioned that you want in addition to the sample itself, you want clinical data
We do.
to accompany it? So
Yeah.
how does one know what you want?
We want as much as you have, really, at the end of the day. But again, this is up to you of how much you want to share with the community. I think more and more patients are open to share as much as there is because they understand the value of something like this. But we do have clinical data and we do have genetic data in our catalog. So we do have several tabs that will have the different information on a sample.
So you ask questions or
We
questionnaires or
send
something?
a form for clinical data in most cases, but we do ask. I mean, the process of us looking at this folder sometimes take months because people will send you a whole folder of everything they've got. So we have to look through it and put the relevant data. But it doesn't matter. The more, the better. The more defined and description that we have for a sample, the better it is.
Thank you.
Well, if that's all your questions, I've been told to send you guys that way for your break and come back at 4:45. Thank you so much for your attention.